as approaching of the cyanobacteria derivatives. So our first approach, why require research? So better medicine than before. So we, the people of the working variety natural products, it's the C2 pharmacy. So comprehensively knowledge about exploration, extraction, screening and trading back compound from the cyanobacteria, their pharmaceutical application will open up new avenues drug discovery and trial prospecting. So if you see that Australia is maximum cover and then the nostric acid, and we are working particularly nostric acid, Australia, and another is the cocoa acid. Most of researchers last 2020, it is updated information that most of people are working with the cytotoxicity, that is cancer-based drug development. Now the era has been changed, WHO has been revised that protocol. So drug has been most required from new variety of antibiotic resistant genes bacterial programs. So we have started our journey with, I have started my journey since 10 years back. It's a full-time devoted work since 2013. We have chosen meeting point of the sea and river, basically Balihastali, Siposargoli, Gabakunda Satkoda, and Gaudiya River Nuha. It's a very beautiful place. If you see this location, here is the river, here is the sea. So on this spotted location, we have isolated 4 strains. That strain basically Coca-Cola minors, Australia himself, Amazonus virus. So we have documented with various review articles where is the research gap in Journal of Biological Structural Dynamics. Hello. So we have documented first isolation of Coca-Cola minors and their antibacterial activity basically in UTI bacteria. So we have did ethanolic extract, acetone, and water extract followed by nanoparticle synthesis, silver nanoparticle synthesis followed by the characterization, then activity of each part. So we have not reached our destiny. Again we are searching our destiny. That destiny again we have searched. So another strain with the MRSA, medicine resistant Staphylococcus aureus, and UTI E. coli. And again we have started with both silver nanoparticles and gold nanoparticles. We have characterized. Then we have not our destiny. We are again first. What is the new era of the research? So it is purely basically on the UTI bacteria. So in here I summarize that cyanobacteria do better than a bioactive fiber compounds. So as per the recent current opinion of biotechnology, it has been reported that the last natural product clinical trial and under the drug approval condition. So is the solitramycin is recently also the natural. Some of the natural product drugs. So cyanobacteria also possesses samples of drugs like rosuvastatin. So these are some cyanobacteria possesses antibiotic drugs. Most of people are working with the cytotoxicity, that is cancer-related works. So we are focusing antibacterial drug development. So another compound which we have been worked with the extract and followed by nanoparticle, we have worked with particularly these Coca-Cola minors, which we have been isolated and followed by nanoparticle synthesis. Nanoparticle synthesis. This is have structurally. I will use this because this Narahan structure possesses from the Coca-Cola minors. It is a beta carbonyl moiety, and we have not isolated with particularly the natural sources. It is already available in sigma in various chemical agents. So we have not wasted our time. So again we have reported even European Journal of Medicinal Chemistry, but it is again gap of the research. So basically we have targeted to position number 9, position number 3, position number 7. So we develop molecule adding of the sulfonamides because our approach is sulfonamide became resistant. So this is another. This is the nostoc. We have also reported is nostocin A. It is a pyrazole moiety. Both two we have one is the isolated from Coccus to Norhaven, another is the Nostrus to Nostrus. So both are reported. So in this prospect, we started semi-synthesis approaches. So we have the problem, again we like the back slide, then that is your disease. So here we have input our advanced computational input, we have the target execution, and our whole work is based on the ligand-based drug design. So our ligand is ready, that is one is the beta-carbolate, is norhaven, and another is nostrosin. So here I am approaching only one for the example. Our target should be bacterial protein, DNA guidance, stopper isomerase. Here I am showing only the DNA guidance. So we will develop lead identification. Then clinical trial. We are under the process of lead identification optimization. We will see next time. So trends of drug development. CAT. Computer-aided drug development is a theoretical model which would utilize lab database to discovery, virtual screening, and newer therapeutic agents. So drug development is an expensive and cumbersome process. Pharmaceutical companies lost huge monetary and due to failure of lead candidates. So moving forward, what actually we are doing. So after our ligand is ready and structure is designed, so this is the literature database. We have our own literature and database. Then our ligand and protein we prepared virtually. Then drug likeness validation, pharmacokinetic study, toxicity assessment, pathway mapping, molecular docking study, drug-disease interactions. So we have reported that in structural activity relationship, in 9th position and 3rd position is most pharmacophore for developing new drugs. Similarly, this is the second nostrosin A also. So it is a Pandora box. We don't know what happens and which molecule will be the lead candidates. So we designed more than 182 molecules based on the mainstream medicinal chemistry principle. After that, speed-based splicing condensation, latest reaction with the high throughput screening. So rule of 5 is basically Lipinski rule of 5. So we further started virtually toxicity, products of that. Then further we have started with our journey with synthesis and characterization. And it is under one condition. So this is the scheme 1. Scheme 1 basically the norhaven, that is the beta-carbolate, which has isolated, we have basically procured, and this is the developed molecule characterized by FTIR, proton NMR, carbon NMR, all these are 2 dynamic NMRs. So, but actual results show that it links to rule of 5, drug stability and absorption, how much our body is taking. Then toxicity we have also, then pharmacokinetic properties. So this is the molecular docking. 1 KZ is basically bacterial protein, and 3G7B is the Staphylococcus aureus DNA guidance. So we have developed these things. So this is the binding interactions of amino acid residue and docking scores with standard drug basically. So dynamic stability of the root mean variation and the root mean square fluctuations. So now we are in ongoing stage. It is already working with now at the laboratory work and institutes, students for the extensive work and the paper plan with the legal trials. And our user generation will be happy there is no drug shortage of the new antibiotics. So in conclusion, natural products have a long track record in antibacterial and continue to be the source in drug installation. This bold promise that natural products revive their earlier success story. If lead optimization enables success in delivery to preclinical and clinical camps, other from the scientific progress, improvements, and commercial ecosystem, funding landscape, strategic partnering, and instrumental to create strong assets and startup to ultimately deliver innovative antibacterial drugs to society. So thanks for giving me opportunity. The Institute of Medical Science and Some Hospital at that time has got ICMR Senior Research Fellowship grant. Vidyasagar University for my master's work from the extension work. CSR for the various part for the characterization, and the great value is a charter company from I have incorporated at the Institute of Life Sciences Bhubaneswar, and reference for the area has been frequently, and she has told that Professor Sarvala Party was she has since worked with me for the Cyanobacteria, and we will be some of the facility for scanning into microscopes on analytical instruments. Thank you.
Thank you, Dr. Siddhartha. Complete the talk on time. So now the topic is open for discussion. So anyone having any questions?
nice talk actually. There is a lot of things you've done actually. Right from starting from this. So, all the docking data that you have shown, yes, this. Hello? Yeah. So, all the docking data that you have done, right? Okay, actually my question was something else. My question was that all the docking program that you have done with some proteins, right? Those crystal structures are already there. And those proteins, did you, means your drug and protein interaction in vitro, did you check it? Okay, so my question over here is, suppose if you are giving it in the bacteria, and the bacteria takes it up. So, if you can check it down before you give to the bacteria and see the antibacterial activity, the protein and the drug interaction. Yeah, the in vitro studies. Yeah, I know. That's why I told you that it's a huge amount of work. That I agree. But diaries, I think you were targeting. You said that the docking you have done in diaries. Yeah, I think topoisomerase is easily, you can purify it. If you put it into a plasmid or something, you can do it. And then you can check the interaction before going to the bacteria. Yeah, anyway, nice talk. Lot of work. Thank you.
Thank you, sir. Now I would like to invite Son Kumar Swami.
Our next presenter is Devi Priyadarshi.
Very good afternoon to everybody. Esteemed chair, all guests from different nations, those who have joined us online today morning, my junior friends, colleagues, my honorable guide there at the chair and everybody else in the hall, a very pleasant afternoon to all. I hope my university has been able to touch upon your appetite nicely. You've had a very nice lunch. Because I'm here to tell you a beautiful story. This isn't any other science like the huge documents which you've been listening since morning from epigenetics to EDTA and whatnot. So we have a little story to tell you from natural history. I am Devi Priyadarshi and I come from the Regional Museum of Natural History, also PhD scholar with the university which is organizing this. I thank the organizers for having given me this opportunity. And I am here to tell you a little about natural history. Now that we have heard so much of technological interventions that science has taken from nature, mostly to apply for the human life though, but we should also learn a little about what nature has as techniques in itself. This is one such classic example when we talk about maternal care in details in a very unique group which is the social spiders. Let me give you a little background. We all know this whole hierarchy of arthropoda, which is the largest in the whole animal kingdom. And if you can see this chart, arachnids in themselves compose for 8% of the whole animal kingdom. And out of that, spiders make up 7%. So once we know all the arachnids in the world, probably this number is still going to increase. So you see this whole amount of insect population there is to be controlled by the rest of the taxa around, of which spiders make an important group that controls the rest of the insects. So classifying their necessity in the animal kingdom, we do not need much details there, but I will be happy to tell you that spiders have been able to colonize almost everywhere in this planet except Antarctica, the cold poles. Coming to the topic, what are social spiders? And like the name suggests, these are not the individual solitary spiders which live on just one web like you must have seen. They are there in your homes, in the forests. Many of you, those who have been nature friendly and walk through lanes or even during morning walk, they come and just dash upon your face. Many don't like the experience though. But there is a group which fall into the family TIASRST, which live in families or colonies. The social structure of these spiders is yet under probe and research. We do not know much about the whole hierarchical status of many in which live inside. But mostly, this is a very small family. Like you can see, there are 100 odd species in the world and just 20 species in this genus Stegodipus, of which we have 4 species in India. One which has been colored in red is actually is an obsolete species which is yet to be proven and has been placed out of this family, although it was discovered once. The 2 in the blue color are the ones which are found in Odisha. One of this, the first one by Foucault 1900, which is Stegodipus pacificus. I will be talking a little about both the species today, but it is a first report in this particular study, which will be from Odisha. And there is just one more stray report, which is there from Odisha, from India, where they report matrilogy in that particular species. So now you must be thinking, what am I talking about so much related to these spiders? You must have seen these structures. Both these structures have not been taken in recent years. One in 2018, the left one, when it was a dew morning in the winters, which is the best time to photograph spider webs. The other one is from Central Indian landscape, where we recorded Pacificus way back in the year 2012. So you can see the structure of the webs and their necessity or the need of such structures is explained well from what they look like. So these are 3-dimensional structures which I have shown you, but mostly what you would find in spiders are 2-dimensional straight structures which are orbs with concentric rings. Now, why they are different? I have written something on the top: triplate and e-triplet silk. Let me walk you through a little about the silk type, which is very, very important when we talk about social spiders. Now, triplet is a small plate which you can see I have marked it here. This particular place is triplet. In a triplet spider, mostly TIASRST belongs to triplet spider groups. They have this cribellum structure which has got fine hair-like structures which it can release along with the silk. When the silk glands actually produce silk, these woolly structures get released from this particular plate. Now the absence of the plate makes it ecribellate or absence of cribellum. So normally spiders would fall into this category, the aranids or the tetragrammatically, say Sorticity. All these spiders would fall into ecribellate category. While few families which are actually thought of to be the old world spiders or more older than the recently discovered ones would have the fully structured silk, which is much better for prey capture. Here we have shown you the live images where you can see the cribellum very clearly, and it is placed exactly at the tip of the abdomen where we have the spinnerets just before that, so that it can aid in producing the woolly structures with the silk. A little more about that. The mechanical stickiness is more with the cribellate silk, like you can see, and when you go into the detailed structures, there is a lot of difference between both. I would not go to the details, making it a little rhetoric of silk itself, but the top picture here on the left, if you can see, this is from family Nephobili, or the fishing net spiders, which are net casting spiders. This is a typical example of what cribellate silk is like. You will never find them straight or with radii which are defined into particular orbs. we will focus into maternal care in spiders. So most of you would already have known that like many other animals in the vertebrate world and also in the invertebrate world, spiders have a very strong history of maternal care. But most of us know that it is limited to only guarding their egg sacs or providing a safe haven for the eggs to be hatched out later into the adulthood or into juveniles. So before the spiderlings hatch, most of the spiders would guard. The off-weavers would tactfully tie them or stitch them into the web and just leave them for the ballooning. But many of them would stick them into the body and wait. You can see the spider here. We have just tried to tear apart a little part of the web, from where you can see that even after the emergence of the babies, the spider is able to guard and keeps guarding till the babies can really release themselves from the nest. Now, so what is so special about maternal care? Because many other animals, we can see maternal care in them. A little closer look to it, you can see these are spiders from the first picture from the cortices, which is a carapace. The other one is from Lycosine family, where they carry the babies on the back as a pouch. All the babies are there cuddled together so that they can keep themselves warm and wait till they can actually release themselves into the wild and fetch food for themselves. Here also we see strong maternal care in courtesy of the jumpers, where we have the babies already hatched out from the eggs, and the eggs, the mother guarding and feeding them with food. We are here to discuss a little detail about what exactly maternal care in the extremities could mean. Now, what do I mean by extremities? Here we are discussing about maternal care where we have such different kinds of stages. There is prey sharing, there is solicitation behavior, trophic egg feeding, regurgitation, and finally obligate maternity. So of all of these, we see many which is available in these kind of families I've already mentioned in heredity, tubularity, heterogeneity, regurgitation we can see in all the erisids and heredity family again. Trophic egg feeding. I would like to tell you a little about trophic egg feeding because this is one method in which the female would actually generate eggs inside the body, inside the abdomen, which can never be laid out. That means, like the oviposition happens, the eggs would usually come out of the body and be laid outside. That does not happen in trophic eggs because they are so huge that it is just meant to be eaten up. And there is a famous thomisid called diet. That is one animal which would eat up the whole abdomen of the mother, including the trophic eggs. We see that in Cheracanthium also, and from Itagidae family, and thomisid. So, let me come to regurgitation now. So, the solicitation I missed. If you can conclude within 1 minute. Okay. Yeah, I need 1 more minute. This is where we talk about obligate maternity. These are some examples that I would like to show you. And because we are in short of time, we will quickly go through it. So, these are the families and the species which we are actually trying to cover up with maternity. You can see here that the babies are actually trying to feed upon. This is a typical social spider nest, and the spider here completely camouflaged some of the insects which are falling prey. And you can see the tunnels here very clearly. This is the egg sac from inside the nest from which the babies have hatched. But later on we found that the dead mother was there inside the nest. That is why obligate maternity is very, very specific to this particular family where the mother would simply lay into a dead sleep. This is the exoskeleton of the mother where it has been sucked out of all the body fluids by the children, I mean the babies, and they slowly grow into juveniles. That is how the life history of this particular spider goes on. What is more interesting is, this is the individual spider which is dispersed out from the colony. What is more interesting is 33% of less cannibalism and mortality has been observed by him et al. In 2001 when the mother was separated and was put together. So these studies tell us that the social construct of these spiders is basically to help increase the social bondage and cooperative prey capture inside the nest. That is very important. And the babies have gained a significant 2.5% weight when they were allowed for activity than otherwise. So what is the takeaway from the study? If this can be used for what my thesis work actually stands for, these are huge machines which can actually capture a lot of insect prey. So if this can be lined all across the paddy fields, it would be dramatic to see how well integrated pest management can actually work. We already had one of the talks on that. So we are yet to prove this, and this is part of the study even now. So we hope we can come back with more results next time. My references, and thank you very much for your patience listening.
Thank you, Devi, for this informative talk. Now I would like to invite some questions from our audience.
Anybody can ask something. We just call them spider legs. We just call them spider legs. You know? No, that is statutorily there. The spider babies are called spider legs. And
So now I will invite Dr. Pallavi Mishra to present her talk.
Respected dignitaries on and off the dais, and dear students. So today I'll be presenting on the topic Eisenia fetidi, the soil nitrification process in the antibiotic contaminated soil. So soil deterioration is one very important global concern nowadays, because the quality of soil, once it deteriorates, then it's very difficult to grow crops, and especially for India, which is an agricultural country. So there are various reasons for deterioration of soil, which we call soil pollution, and there are many factors for that, like bad agricultural practices like crop burning and applications of different agrochemicals, be it fertilizers or pesticides, insecticides, which go into the soil. So, and apart from that, heavy metals, microplastics, all these things. But one aspect which has been not researched upon and neglected area of soil pollution is antibiotic pollution. So as we know that antibiotics which are given to human beings and livestock animals to prevent bacterial diseases, these antibiotics make their way into the soil through various sources. Maybe through hospital effluents or through the fecal matter, because there is open defecation in India, which is a major problem, or by burning of carcasses, or through sewage water, which is used for irrigation purpose, and by application of manures. So as we know that now we are more interested in organic farming, so we are applying animal manure as fertilizer. But these antibiotics which are given to these animals, they are not properly, if they are not properly degraded inside the gut, then these antibiotic residues will be dropped into the soil through their fecal matter or through their urine. So once these antibiotics make their way into the soil, they will make with subsequent application, these will make the bacteria present in the soil resistant. And another important factor is that these antibiotics may be through the flood water, they may enter into the water bodies, and they may also enter into our food chain. And apart from that, another problem is that they will also change the bacterial community in the soil and degrade the bacteria which are responsible for the nitrification process. So these antibiotics antibiotics, there are some broad spectrum antibiotics like ciprofloxacin, tetracycline, which have a very large absorption coefficient and long life, so they will stay in the soil for many days and gradually they will make the soil bacteria resistant. So we are working to find one of the solution for this with the help of these humble creatures. Which we all know that these are also known as the ecosystem engineer or the farmer's friend or the soil remediator, which are the earthworms. So our hypothesis is that when these antibiotics, when they are contaminating the soil and they are passing through the gut of these earthworm, because soil is a food for earthworm. So in the earthworm gut, there are millions of microbes and enzymes which will process or degrade these antibiotics. So when these soil contaminated, antibiotic contaminated soil is passing through their gut, it will be either antibiotics will be degraded or they will also remove the antibiotic resistant bacteria and subsequently the genes. So with this hypothesis in mind, we have developed this experimental design where we have taken two types of soil for an experimental period of 28 days. In one, we have applied the antibiotics along with the earthworms, and in another, we have applied only the antibiotics. And we measured different parameters after 28 days. Like we measured the concentration of the antibiotics by HPLC, then we measured the abundance of the antibiotic resistant genes by quantitative PCR, and also we did the sequencing to know the bacterial community structure. Apart from that, we also wanted to see that how the nitrification process is affected. So we also did measured the ammonium nitrogen and the nitrate nitrogen, as well as the ammonia oxidizing bacteria genes and the nitrate oxidizing bacteria genes. Because nitrification process has two steps. First, the ammonia is converted to nitrite by these ammonia oxidizing bacteria, and then this nitrite is converted to nitrate by this nitrite oxidizing bacteria. So we wanted to measure their abundance after these antibiotic applications. So these are our results. This is the HPLC results, and we found that the concentration of antibiotics is getting lower when they are in the presence of the earthworms. And this is the graph showing the ammonium nitrogen and the nitrate nitrogen content, and as you can see here, in both the cases it is the earthworms are able to remediate that. And this is the qRT-PCR data of the AOP specific genes and the NOP specific genes, and these are also improving. So with this I like to conclude that these antibiotics, they are degrading the soil quality by inhibiting the nitrification process, and earthworms with the help of their gut-associated processes are able to remediate them. But one important problem is that earthworm also faces if these antibiotics and pollutants, they also harm the earthworms. And second is that the microbes present in these earthworms, they are very few in number in comparison to the microbes present in the soil environment. So if we can protect these microbes, if we can give them a microhabitat, so they can flourish in the soil environment. So thinking this in mind, we are trying to find a carrier system where we can inoculate these microbes from the earthworm and put them in the soil. So we took this biochar. Biochar is a carbonaceous product which is formed by heating them in low oxygen, and this is the charcoal-like product which we get, and as we are getting it from biological waste, so we call it biochar. So biochar also has its own advantages, like it helps in mitigating climate change, then it also improves the soil quality because it itself has nutrient quality, so there are different aspects of that. So we took biochar and then composted, and then we inoculated the microbes which we had isolated from the earthworm, and we inoculated that with this biochar. So now we have provided them with a home, with a microhabitat, so that they will flourish without any competition from the soil microbes. So because these biochar has so microbes can inhabit this biochar and be present in the soil. So this is the application part where we have taken coconut shells because these are readily available where I work in Puri, and these coconut shells have been converted to biochar, and we have inoculated these biochar, microbes from the earthworm inside this biochar, which we call as vermi biochar. So this is the application part. So this whole project, this work has been funded by ODIP Seed Fund, which is under Government of Odisha, and we have filed an Indian patent application for this biochar. So with this, I'd like to end my talk. These are the references, and at last, I'd like to thank my college, that is SCS College Puri, for giving me the lab infrastructure to do this work. And finally, I'd like to thank Department of Zoology Utkal University for giving me this opportunity to present my research work in front of you. Thank you.
Thank you, Dr. Devi, for the new and the environmental benefited talk. So the topic is now open for discussion. Anyone having any query?
Yes. So actually we also work on biochar and your dye remediation. I just have one question. So when you are giving the biochar, supplementing it with the soil, the biochar has the ability with the functionalization. I don't know what functionalization you're doing.
Yeah, that part we are working now. How these, what are the characteristics of this biochar, how it is maintaining the pH, how the anion exchange capacity. That we are working. The earlier part has been already done. So now we are working on how this biochar is maintained in the soil environment and for how many days it is maintained.
No, actually the antibiotic that you are using, the biochar also has the ability to absorb that.
Yes, yes, yes.
So it is like two mechanisms acting over there.
Yeah.
One is the earthworms, whom you are telling that they are taking it, and the other is being absorbed by the biochar.
Yeah, biochar itself can absorb the antibiotic, and we are also giving a microbes and enzymes from these inoculated. So there's a nitrification process is also maintained along with that.
Okay, so you have a control experiment where biochar only how much it is absorbed?
Yes, yes.
You know that. Okay, and one more question is that you showed a HPLC graph, two of them, right? So do you see any degradation product also after the ingestion happens?
Sorry, I was not able to.
Like suppose an antibiotic X, after it is eaten up by your
Yeah, we had two parameters. One was only the antibiotic contaminated soil. We took out the antibiotics and measured it, and then another where the earthworms were present, and we also measured that.
So do earthworms only absorb it or they break it also?
They break it also.
They break it also. So we do observe the products of the breakup also in the HPLC?
Yes, yes, but there was not much quantity.
Okay. Okay, got it.
Thank you.
Thank you.
Now I would like to Ms. Jasmine Rao to present her talk.
My topic is cyclones affect the heterogeneity and richness of amulans in Eastern India. I am Jasmine Rao from Department of Utkal University, and amphibians is coming from two Greek words. Amphibians dual and bios means life. So they have dual phase of life. One part exists in water and another part exists on land. So biologists believe that amphibians appeared during the late Ocean period, about 345 million years ago. So this is the classification of amphibia. Amphibia is divided into two extinct, is divided into one extinct subclass and another living subclass. Extinct subclass Tecocephalia was divided into three orders. One is Labyrinthodontia, second one is Thalospondyle, and the third one is Rhymphospondyli. Labyrinthodontia is coming from one big word labyrinthodont, which means bend tooth. Another example is Rhymphospondyli, and the second spondyli, which was also a Greek word, phyllospondyle, which means poor ossified orthopod. The best example was Brachiosaurus, and the third one was Lebecospondyle, which is the true ancestors of modern amphibia. And the living list amphibia is divided into three living orders. One is Anura, second one is Chordata, and third one is Gymnophiona. Anura including frogs and toads. Frogs and toads are available in all over world except the polar regions, some desert regions, and some isolated islands. The second one is Urodella, the modern name is Chordata, which includes salamander, and the third one is Apoda, means without leg, which includes Sicilians. So the amphibia wave database currently lists 8,579 amphibian species, of which Anura share 88%, Chordata share 9%, and Sicilians share only 3%. And the highest number of species is found in the family Hylidae, and followed by Chapomonidae. India is home to 42 species, out of which 306 belong to the order Anura. And in India, family Rhacoponidae has maximum number of species, followed by Rhacoponidae. Globally, the amphibians are more created as compared to other groups of animals, with approximately 48% of more than 8,000 amphibian species. So these are some of the anurans which are recorded from Odisha according to Datta et al. 2009. So why should you go for conservation research on amphibians? Amphibians are very important for us as they are exploited as food sources in some countries. They have the medicinal properties. They are used as laboratory material in many schools and colleges. They are the important constituents of food chain. So there are many regions of declining of amphibians, out of which habitat degradation, habitat conversion, habitat fragmentation, fertilizer pesticide stressor, deformity, introduced species, diseases, UV radiation and over-harvesting. And last one is climate change and natural calamities. Natural calamities such as cyclonic storms can affect the adult anurans as well as larval amphibians in many ways and could be the major cause of amphibian deterioration. So India experiences large number of cyclonic storms in every year, but the effect of cyclones on amphibians is very less. The study of effect of cyclones on amphibians is very scarce. So my objective is to compare the amphibian species richness, abundance and composition among the different habitats. And the second one is to determine the effect of cyclones on species richness and community composition. So these are the study sites of Eastern Odisha. So for this study, we surveyed for both adult and larval anuran before and after the cyclones. There are 9 major cyclones happened during our study period, but we focused on only 3 cyclones. That is Fani, Amphan and Yas, because their intensity is higher in comparison to other cyclones. So our data were partitioned into periods of pre and post cyclones, and the categorizations are as follows. We consider pre-Fani period from 1 May 2018 to 26 April 2019. And post-Fani or pre-Amphan period is from 6 April 2019 to 16 May 2020, as Fani occurred in Odisha on 3rd May 2019. According to that, post-Amphan and pre-Yas period from 22 May 2020 to 23 May 2021, and post-Yas period is 29 May 2021 to 31 January 2020. So 3 survey layers were used to acquire anuran abundance by conducting visual encounter surveys, including refugee examination under holes, under trunks, and diagonally and nocturnally each side, and each survey lasted approximately 1 hour. So survey data were organized as counts of individual per species, per habitat and per year. For local unanswered three types of study area, we used one is permanent pond, second one is temporary pond, and another one is canal. So animals that could not be positively identified in the field itself, they are collected and identified in the laboratory, and those individuals were returned after the identification. So during each survey, we recorded the environmental parameters, and environmental parameters are important for amphibian breeding conditions. So the environmental parameters pH, air, water temperature, water depth, relative humidity, canopy cover, and summer nutrition were measured during the study period. So this is the result. So we analyzed the adult anurans in marsh and swampy habitat condition. So Datapranus melanostictus, Eucalyptus sanophlictis, Phaedovaria odyssensis, Haplobotrachus crassus, Haplobotrachus tigerinis, and Polyphytes maculatus, Microhala ornata, Microhala rubra, Eupredon globulosus are found in both marsh and swampy condition, but Datapranus domesticus, Sparatheca breviceps, Polyphytes terrensis, Calola taprobanica, Eupredon systema is only present in swampy habitat condition. So, basing on community composition for marsh and swampy habitat, Polyphytes maculatus constituted 52% of total community in marsh condition before funny, and in swampy condition, Calola taprobanica constituted 36% of total community composition, followed by Polyphytes maculatus. This is the condition of pre-funny period. So, after funny, the Haplobotrachus crassus constituted 27% of total community composition, whereas in swampy condition, Polyphytes maculatus constituted the highest number of community composition. So the scenario got totally changed. So in post-amphibian condition, Microhala ornata constituted most of the community in marsh habitat, whereas in swampy habitat, Polyphytes terrensis constituted the highest number of community. So in post-years condition also, Polyphytes maculatus constituted for 23% of the community composition, whereas in post-years condition also, in swampy region, Polyphytes maculatus constituted 20% of the composition. So here, if we see the pH concentration in 4 cyclonic periods, after funny, the sudden increase of pH might be the cause of declining of anurans in eastern Odisha. So this is the percentage of anuran occupancy of total water bodies for egg laying during the study period. You see, in pre-funny condition, permanent pond constituted highest number of tadpoles and egg clutches. But after funny, the number got decreased. So this is the breeding preference and polynomial trends within different ranges of pH. You see, in highest pH concentration, we found this number of tadpoles. Marsh habitat constituted or recorded highest species richness, highest species diversity, whereas in swampy habitats, species richness is more than diversity. So the cyclones change the species diversity and evenness within each habitat, which could have an impact on long-term community dynamics and species interaction. Therefore, extrinsic insults like powerful cyclonic storms impact anurans in various ways in recipient regions of the world. The effect of cyclonic storms on anuran in coastal habitat as well as in other part of the Odisha, other ecosystems affected by alkalinity may be more severe than anticipated, and it is likely to have comparable effects on other ectoderms. This knowledge has obvious benefits to management and conservation of valuable habitats that are used by amphibians. So these are my acknowledgements, references.
Thank you, Jasmine. So this is the last talk in last presentation of the conference. So anyone having any questions? Query?
protect the amphibians from the ill effects of the cyclone?
My PhD topic was decimal ecology of amphibians. So in our study area, I was recorded the species diversity and species richness. So after cyclone, suddenly I noticed that the species number got decreased. So
Yeah, I understand that. But is there anything we can do to help the amphibians, to protect them when we know that there is a cyclone coming?
actually, my study
That was a really good presentation. I have one short question. What do you think? I know that you have not done this research, but can you speculate how much time it will take to recover, go back to the original state of diversity? So cyclone disturbed the habitat, but will it have to, maybe perhaps it will either recover and go back to the original stable population, stable dynamics, stable diversity, or does it lead to all new species diversity? Do you have that speculation?
So actually, till now, it is not recovered to the original condition. But in our study, we noticed that after funny, the species number and species diversity, species number is got decreased after funny. But lateral in our analysis, we observed that the original species number and species richness certainly slowly got increased after funny.
Thank you, Justin. Now, I invite all four speakers come on the stage so we can felicitate you.
So may I request our chairperson, today's last meeting, Dr. Jostarani Pradhan, to distribute the certificates to the participants. Madam, please.
लाइट अभी लगाऊंगी लाइट आ गई
Many thanks to Dr. Pradhan for chairing this session. Thank you, ma'am.
Thank you, ma'am, for giving me this opportunity to chair this session. Thank you, the organizers.
And now we will get into the last part of to organizing secretary for rest of the events.